For over a century, scientists believed our ancient world was home to just two human kinds, until a 12mm finger bone from a Siberian cave exposed a lost branch of humanity. This DNA rewrote human origins, linking a Tibetan jawbone found on a high plateau to a species still shaping modern genes. What else have we missed about the Denisovans, and how far did their legacy really reach? In 2008, deep inside Denisova Cave in the Altai Mountains of southern Siberia, archaeologists uncovered a tiny fragment of bone just 12mm long. It looked unremarkable, a sliver from the pinky finger of a young girl who had died tens of thousands of years earlier. Yet this fragment would soon upend everything scientists thought they knew about human origins. The bone was carefully packed and sent to Leipzig, Germany, where geneticist Svante Pääbo and his team at the Max Planck Institute for Evolutionary Anthropology prepared it for analysis. Their specialty was extracting ancient DNA from the most unpromising remains, a process that demanded both technical precision and luck. The team began with mitochondrial DNA, which is more abundant and often survives where nuclear DNA does not. As the sequences came in, something astonishing appeared. The genetic code from this finger bone was unlike that of modern humans or Neanderthals. It branched off nearly 1 million years ago, forming a distinct lineage, one that had never been seen before. The results, published in Nature in 2010, confirmed that a third kind of ancient human had lived alongside Neanderthals and our own ancestors. The scientific world now had a new name to add to the human story: Denisovans, after the cave where the bone was found. The implications were immediate and profound. While Neanderthals had left behind hundreds of bones and even partial skeletons, the Denisovans were known only from scattered fragments, a finger, a few oversized molars, and later a partial skull. Their physical appearance remained a mystery. To fill the gaps, geneticists turned to DNA methylation patterns, chemical markers that influence how genes are expressed. These patterns suggested Denisovans had dark skin, brown hair, and wide faces, but the evidence was indirect. The reality was stark. An entire branch of humanity, identified first not by bones or artifacts, but by molecules, recovered from a tiny piece of bone. Pääbo's breakthrough revealed that Denisovans were not a minor offshoot. Their DNA showed remarkable variation, even more than all sequenced Neanderthals combined. Modern genetic studies would soon find traces of Denisovan ancestry scattered across Asia and Oceania, but at that moment the world had only a genetic signature and a handful of bones. The question loomed: what did these people actually look like, and how far had they spread? The search for answers had only just begun. In 1980, high on the Tibetan plateau, a Buddhist monk searching for relics in Baishia Karst Cave stumbled upon a heavy, weathered jawbone buried in the sediment. The cave sits at 3,280 meters above sea level, where winter air burns the lungs and even modern visitors struggle for breath. The monk handed the jaw to local authorities, who placed it in a museum without suspecting its true age or importance. For decades, the fossil gathered dust, its story untold. Nearly 40 years later, researchers from Lanzhou University, joined by scientists from the Max Planck Institute, gained permission to study the ancient bone. Hopes for DNA were quickly dashed, because the cold, dry conditions had not preserved any genetic material. Instead, the team turned to a different molecular clue: proteins, the resilient building blocks of bone. Using a technique called collagen fingerprinting, they extracted and sequenced tiny fragments of protein from the jaw's mineralized core. Collagen, unlike DNA, can survive for hundreds of thousands of years, holding chemical signatures that point to evolutionary relationships. The results were unmistakable. The collagen peptides matched those of Denisovan fossils from Siberia, confirming that the Shiahi mandible belonged to the same enigmatic lineage. Dating of a crust of carbonate on the bone placed it at least 160,000 years old, making it far older than the famous Denisova finger bone. This was the first Denisovan fossil ever found outside Siberia, and the first from a region so high above sea level. The jaw's robust shape and thick bone hinted at a powerful build, but the real revelation lay in its location. Denisovans had not only survived at extreme high altitude, they had thrived there long before modern humans arrived. Later studies would show that Tibetans carry a gene for high-altitude adaptation inherited from Denisovans, helping them flourish where oxygen is scarce. The Xiahe mandible forced scientists to rethink the Denisovan story. Here was proof that this ghost lineage ranged far beyond the Altai Mountains, adapting to some of the harshest environments on Earth. Yet even with this breakthrough, the Denisovans remained faceless, known by fragments, not by a complete skull. The search for a true Denisovan face would lead researchers further east and deeper into the puzzle of our own origins. In the summer of 1933, a laborer working on a bridge near Harbin in northeast China unearthed a skull unlike any he had ever seen. Fearing it would be seized by occupying forces, he wrapped it carefully and lowered it into an abandoned well, where it remained hidden for nearly 90 years. Only after his death did his family reveal the secret, handing the skull to scientists at Hebei Geo University in 2018. The cranium was massive, with a wide face and thick brow ridges, features that sparked fierce debate among researchers. Some argued it was a new species, Homo longi, while others saw echoes of archaic humans from the icy caves of Siberia. The question hung in the air: who was Dragon Man? For years, the answer eluded researchers. The breakthrough came not from the bone itself, but from the thin crust of dental calculus still clinging to its ancient teeth. In 2025, a team of geneticists and proteomics experts at Hebei Geo University and the Institute of Vertebrate Paleontology and Paleoanthropology set out to extract mitochondrial DNA from this hardened plaque. The process was painstaking. 20 samples were tested, and only 7 yielded usable genetic material. Each step was checked for contamination. The results were unmistakable. The mitochondrial DNA from Dragon Man's calculus clustered tightly with Denisovan specimens from Denisova Cave, especially those over 100,000 years old. At last, decades of speculation ended. The Harbin skull was Denisovan, the first complete cranium ever linked to this mysterious lineage. For the first time, scientists could study the face of a Denisovan, not just its genetic shadow. What began as a laborer's act of quiet defiance became the key to solving one of paleoanthropology's greatest mysteries. The discovery gave a face to a long-hidden story and reshaped our view of human evolution. Sediment DNA is rewriting the Denisovan map. In caves across Asia, from the Tibetan plateau to the humid forests of Laos, scientists have recovered ancient genetic traces left behind not by bones, but by fragments of DNA preserved in soil. At Baishia Karst Cave in Tibet, layers of sediment yielded Denisovan mitochondrial DNA spanning more than 50,000 years, confirming their repeated presence at high altitude. Further south, in Tam Ngu Hau Cave in Laos, a Denisovan tooth and protein analysis point to their reach deep into Southeast Asia. Even where fossils are missing, sediment DNA reveals that Denisovans once moved through these landscapes. The story grows more complex with the discovery of a hybrid child in Denisova Cave. A small bone, dated to about 90,000 years ago, belonged to a girl whose mother was Neanderthal and whose father was Denisovan. This single individual stands as direct evidence of interbreeding between archaic human groups, a genetic crossroads that shaped the DNA of future generations. Denisovan legacy is not just ancient history. Modern populations in Melanesia and parts of Oceania carry up to 6% Denisovan ancestry, the highest known today. In Tibet, the EPAS1 gene, essential for surviving low oxygen environments, comes from Denisovans and allows people to thrive at extreme elevations. These genetic signatures are living reminders that Denisovans are not just a chapter in prehistory. Their DNA continues to shape the biology of millions across Asia and the Pacific, connecting us to a vanished world mapped by molecules in the earth and in our own cells. Today, Denisovan DNA shapes the oxygen in Tibetan blood and lingers in millions of living genomes. As ancient molecules redraw the boundaries of humanity, one reality endures. Our origins are still being written, and every new fragment can rewrite who we think we are. Share your thoughts below.